Now we're going to discuss active transport. So this is our third type of cell transport. And so if you didn't draw a little squiggle right above this, let's draw that little squiggle, which will help us divide between facilitated diffusion and osmosis, which was our second type of transport, and active transport, which is our third type. So for active transport, this is going to require energy. And because it requires energy, it's going to help us move things from areas of low concentration to high concentration. So the opposite of our passive transport, which does not require energy and goes from high to low. So active transport is helping substances move against the concentration gradient. So how do we do this? We do this using a protein pump. So it's still a protein, which is embedded into our cell membrane. But in this case, this protein is going to require energy from ATP. Remember, ATP is energy that our cells can use. And this helps us move small molecules and ions across the membrane. And again, this is going to go against the gradient, so from low to high. And how proteins do this is they change shape in order to help pump the molecules across. So if we take a look at this protein pump, and we take a look at our pictures here, what I can see is inside the cell, I have some molecules that need to be carried, and outside the cell, I have some that need to get out. Now, I actually don't like this picture because this picture doesn't make it super obvious where the high and low concentration gradient is. So what I want us to do is I want us to draw in some more solute things. So I'm going to draw in some more circles on the outside so I can make this picture true to what we want it to be. So add in some circles. Because as you can see in the picture, the particles are moving from inside the cell to outside the cell. And if this is a protein pump, it's supposed to be moving things from low to high. So I just want to make the picture be true to that. So let's look at some ways that cells can bulk transport things into and out of the cell. So first, let's look at endocytosis. Endocytosis, I want you to focus on the endocytosis, is how cells take large materials into the cell. And just like we talked about when we were discussing proteins moving throughout the cell, our mode of transport is going to be using an organelle called a vesicle. The opposite of endocytosis would be exocytosis. And in exocytosis, this is how we release large molecules out of the cell, again, using a vesicle. And we showed exocytosis when we were following that path of the protein from the ribosome to the rough endoplasmic reticulum to the Golgi body and eventually out of the cell. Exocytosis was a way that the cell does this. So over here in the picture, you can see endocytosis. We have the molecule outside, and what happens is the cell membrane helps form this vesicle, which then takes the molecule into the cell. And in exocytosis, this is where the membrane surrounds and fuses with the cell membrane, and therefore we can force the contents out of the cell in exocytosis.